actual genes and mutations that contribute to adaptation? If the same phenotype evolves, is it because it's due to changes in the same genes and even the same mutations, or completely different sets of genes involved? And the question that I'll really focus on today is, does sort of co-inheritance mediated by genetic linkage and or pleiotropy facilitate adaptation or actually act as a constraint on adaptation? And like I said, this is the focus of my talk today, but I will touch on some of these other issues as well throughout the talk. So as she mentioned, I answer these questions using the Threespine stickleback as a model system in order to connect genetic and phenotypic variation. So just a brief background on sticklebacks. They are a small teleost fish. They live in very diverse habitats, so they live in both the ocean, some marine habitats, and then in freshwater lakes and streams. We see really extensive phenotypic variation both between marine and freshwater environments, and then also among freshwater environments as well. What's really nice is we have these sort of natural replicates, so marine sticklebacks have invaded freshwater thousands of times over, and then we also see replication across freshwater habitats, which really gives us the opportunity to ask, what are the same genetic changes involved when similar phenotypes evolve? What's really great for me as a geneticist is that I can take these very divergent populations, I can bring them back to the lab, and I can cross them and generate lots of offspring. Not anywhere near as close as you can in Drosophila, but we can get good numbers of offspring. And of course, we have now some genetic tools, and as we all know, it's super easy to sequence the genome. And the genome of sticklebacks is only about half a gigabase, so it's actually a pretty compact vertebrate genome. Okay, so a little bit more about the evolutionary history. The ancestral populations we think is marine populations, and about 15,000 years ago, as the glaciers melted, these marine populations were able to invade newly created freshwater lakes and streams across the northern hemisphere. And so we get these derived freshwater populations sort of that have diversified again across the range of sticklebacks in both the Pacific and Atlantic basin. So again, we have these repeated adaptations. So the first part of my talk is going to focus on differences between marine and freshwater sticklebacks. So these are just male sticklebacks tending their nests with their beautiful blue backs and red throats they use to attract females. So one of the most obvious differences between marine and freshwater sticklebacks are these bony lateral plates that I'll talk about a little bit. So these fish are stained with alizarin red to highlight the bone. So marine sticklebacks have this set of bony armor, bony plates that go along their side, and also these big spines on their back and spines on their belly. However, most freshwater populations have lost these posterior plates, and so they just have these plates here in the anterior of the body. But marine and freshwater sticklebacks vary in all sorts of traits, and so pretty much everything we can look at, so body size is a big difference. Marines tend to be larger. They have this much more silvery pigmentation than these sort of stripy or mottled stream resident sticklebacks. Behavioral differences, so marine sticklebacks are migratory. They move into freshwater to breed, and then the babies are born in freshwater and then migrate back out to the ocean, much like salmon. Whereas freshwater resident sticklebacks spend their whole life in freshwater and in fact have lost the physiological ability to live in salt water. We've also worked a lot on schooling differences, so marine fish school very strongly, freshwater sticklebacks don't. And then there's also some life history differences. But these are just some examples of the many, many phenotypic differences between marine and freshwater populations of stickleback. Okay, so as I mentioned, though, we can cross these very divergent sets of populations in the lab and get viable and fertile offspring. And this allows us to use quantitative trait locus or QTL mapping, in which we basically establish crosses in the lab between divergent populations. We make F1 hybrids, we intercross those F1s to make an F2 population in which we basically shuffled the genomes of the starting populations, and then we can score these F2 generation. We look at whatever traits of interest. So, do they have plates or not? Do they have a pelvic spine or not? The length of spines. And then we can just do genome or QTL mapping. We look for associations between genotype and phenotype. And then ultimately we can use these QTL to delve into these genomic regions and ask, well, what genes are in these regions that are associated with differences in phenotypes. And ultimately our goal is to identify molecular changes that underlie these interesting phenotypes. Okay. And so this sort of work QTL mapping step has been done a lot in Stickleback. And together with David Marquez, who was a PhD student with Ole Seehausen at the University of Bern, David had compiled a big database of 28 QTL mapping studies that had been done. And these QTL studies, there was over 1,000 QTL in this database. And David and I put them into different trait categories. So there's a lot of morphological traits. So we sort of associated those with feeding structures, defensive structures like those plates or spines, which are thought to be defensive against predators. There's a lot of shape variation, so that people had measured. There were some reproductive traits, and then also a number of behavioral traits. And so with this big data set, David and I went back to these specific questions that I posed, again, at the beginning of my talk and tried to address some of them. And again, I'm not going to focus on the answers to these other questions. You can ask me about them in the question and answer period. But the one question we really wanted to know was, does genetic linkage or pleiotropy facilitate adaptation? So do we see that QTL are sort of distributed across the genome? Or do we see that they're sort of clustered in the genome, which would suggest that sort of linkage of traits of different traits or pleiotropic effects of single mutations on multiple traits might actually facilitate adaptation. And so the result of that analyses they're shown here. So on the bottom are the 21 different stickleback chromosomes. And then we've sort of looked at all of the QTL together. And then for the categories of feeding and body shape and defense, we had a lot of QTL. And so we could sort of look at the distribution of specific types of traits. And then what I want you to focus on is just looking at sort of for each chromosome, are there lots of sort of a high set of bars or a low set of bars? And so the first result is if you look across all QTL, there's QTL on every chromosome. So there is somewhat of a distributed genetic architecture that QTL can be sort of everywhere in the genome. But we do find that there are specific chromosomes where you find more QTL than we expected, either given the number of genes on that chromosome or just the total length. And so chromosomes 4, 7, 16, 20, and 21, these cluster, where we find these clusters of QTL. Interestingly, 2 of these clusters on chromosome 4 and 21 are found sort of across all sorts of trait categories, whereas the cluster on chromosome 7 seems to be more associated with defense traits. This cluster on 16 is associated with shape traits, and this cluster on 20 is associated with feeding traits. So there's sort of a breakdown there. Okay, so this suggests, yes, there is some clustering of traits, and so maybe co-inheritance of traits might facilitate adaptation. But the question that we really wanted to ask then is, well, how are these clusters formed? Are they due to a single mutation that has pleiotropic effects on different phenotypes, so multiple morphologies or behavior, physiology? Or is it that there's linkage of multiple mutations, and so each of these mutations kind of separately affects different traits? For example, one mutation affects morphology, another behavior, another physiology. And at the level of the QTL, I mean, we have big regions of the genome, are found in these QTL. There's usually hundreds of genes. And so it's sort of really hard to address this question. And, most studies that find this cluster genetic architecture sort of stop there and say, okay, yeah, we find this clustering. Is it pleiotropy or linkage? That's the question in the discussion. But we wanted to try and address that question. So we honed my PhD, or former PhD student in my group, Sophie Archambault, she defended about a year ago, set out to address that question in her PhD. So Sophie focused on this QTL cluster on chromosome 4, about which we knew sort of a lot. We also knew that on chromosome 4, in this work of Felicity Jones, where she sequenced 10 marine and 11 freshwater populations and then looked for regions of genomic divergence between marine and freshwater populations, that one of these sort of 80 regions that she found was on here on chromosome 4. So to just hone in, she found that there's a 16 kb haplotype in which marine fish sort of have fixed alleles that are different from all the freshwater fish. And this is, again, from around the globe, okay? So we knew that there's this really strong regions of genomic divergence between marine and freshwater within this sort of QTL cluster on chromosome 4. So what's in this haplotype? It has 3 genes. This gene called ectodysplasin, or EDA, which is a secreted signaling molecule that's involved in the formation of ectodermally derived tissues like scales in fish. Sticklebacks don't have scales, they have those plates. It's also involved in the formation of feathers in birds and hair, teeth, and sweat glands in mammals. These other 2 genes we know less about, but TNSF13B is thought to be involved in B cell function. And this gene GARP is also thought to be involved in immune function. Okay, so there's 3 genes. There's also 17 fixed polymorphisms between marine and freshwater, so both SNPs and small indels. So again, if we sequenced a marine fish from around the globe, we would find that it carried sort of marine alleles across these 17 polymorphisms, whereas freshwater fish would sort of have the alternative allele across this haplotype. Okay, so we know a bunch about the function of at least one gene in this haplotype EDA. So previous QTL mapping studies followed by functional studies had shown, in both David Kingsley's group and my group, had shown that the EDA gene is expressed at higher levels in the marine sticklebacks than in the freshwater sticklebacks. In this case, we use this benthic population. And if you overexpress EDA in a freshwater stickleback, you find that that can basically drive the formation of plates. So EDA is responsible for this difference in lateral plate phenotype between marine and freshwater. My group showed that it's also involved in a difference in the sensory hair cells called the neuromass, which is this sensory system that aquatic vertebrates have to sort of sense water motion. So on these plates, we see that marine sticklebacks have these sort of pairs of these neuromass, these little dots, whereas freshwater sticklebacks in unplated regions have sort of a single line of neuromass. And again, overexpression of EDA in freshwater sticklebacks drives the formation of this marine-like pattern in the neuromass. And we also showed that leads to a difference in schooling behavior. So marine sticklebacks school very strongly. Freshwater sticklebacks sort of tend to not school very strongly or not maintain a parallel position in a school. But overexpression of EDA in freshwater sticklebacks drives them to behave more like marine sticklebacks in this schooling assay that we developed. Okay, so we know that the EDA gene itself has pleiotropic effects on at least 3 phenotypes. But what we didn't know is whether that was due to a single mutation, a pleiotropic mutation that was sort of affecting these different phenotypes, or whether this was due to linked mutations that each independently affect these different phenotypes. We also didn't know if the sort of QTL cluster with all of these traits sort of mapping to the center of chromosome 4, if any of these other traits were driven by EDA. And so to address that question, we really wanted to do some finer mapping. So we wanted to generate lots and lots of recombination across this region. So we could ask, well, what are the specific mutations in this 16 kb region that are associated with these different phenotypes? So if I was a Drosophila geneticist, I could do this all in the lab, right? I could generate tons and tons and tons of flies and look for those super rare recombinants. But we don't have enough fish space to do those sorts of experiments in the lab, and sticklebacks have a 1-year generation time. So we had to take an alternative approach to this recombination mapping in the lab. And then, of course, ultimately, we want to validate those things with functional experiments using CRISPR. Okay, so what did we do? We instead took an association mapping approach. So we had a population that we had studied extensively in my group in Seattle, this big lake, Lake Washington. A former postdoc in my group, June Catano, had shown that the sticklebacks in Lake Washington are actually polymorphic. So it's a freshwater lake, but they're polymorphic for this plate phenotype. So you can find fish that are completely plated, more like a marine phenotype, partially plated, and low plated. And their genotype at EDA is, I'm now going to switch from sort of calling it the marine genotype, and they're sort of homozygous for the marine allele. We're going to call it CC for complete. These partially plated fish are heterozygous for the marine and freshwater allele, and they're CL. And the low plated fish have the 2 freshwater forms of the haplotype, and they're the LL genotype. So I'm going to use these CC, CL, and LL genotypes. But besides varying at plate phenotype, essentially this is a panmictic population. So at other markers, they seem to be sort of freely interbreeding. So this provided a perfect opportunity to ask, could we find recombination across this across the EDA locus, and then use association mapping in this population to see what allele was most strongly, or what mutation in the haplotype was most strongly associated. So again, we would just collect fish, phenotype them, and then genotype them with markers across the haplotype. And if there's recombination, we could sort of see haplotypes that are now a combination of, freshwater alleles at some markers and marine alleles at other markers. And then at every marker, you just do an association test and ask where is the strongest association. So this peak here would suggest that the mutation that drives plate number, for example, is somewhere in this region of the haplotype. Okay, so we collected close to 1,000 fish over 2 years, and then genotyped those fish with markers across the haplotype, and then phenotyped them for all the traits that we knew mapped to this bigger cluster on chromosome 4. This field work is not always like super pleasant. Sometimes it's really muddy, and it's usually raining too, especially in Seattle. Okay, so the first result that was exciting is that we had actually found evidence for recombination across this haplotype. So most of the fish that Sophie genotyped were non-recombinant. So here again are all the markers across this region, and a lot of fish were they had LL genotypes across all of the markers. A lot of the other fish were heterozygous across all the markers, and then there was fish that were homozygous for the C allele across all the markers. The interesting fish were these close to 200 fish which were recombinant. So for example, each line here is an individual fish, and the colors sort of indicate the genotypes. So for example, this fish has LL genotypes all the way across the haplotype, and then there's been recombination events between these sets of SNPs, and now it's CL here. And these are the fish that are super interesting to us. They're the ones that allow us to sort of do the genotype-phenotype mapping at each marker. And I'll just cut straight to the results. Oh, no, sorry, I won't cut to this. First, I need to mention that I was talking to Christian about this earlier. The phenotyping is actually a huge amount of work. So Louis Berchi, for his bachelor's thesis in my group, phenotyped these fish for 124 total traits, 41 of which had been previously mapped to this cluster on chromosome 4. So these were these lateral bony plate phenotypes, these neuromass or lateral line phenotypes, and then all sorts of morphological phenotypes that we could measure. Importantly, it's just morphological traits. And then here's what we found. So here are sort of the positions of markers within the haplotype, and then on the y-axis is the LOD score. So this is just the strength of the association between genotype and phenotype. And so for the plate count, so whether the number of plates, we see that the peak association is somewhere here between these two length polymorphisms or small indels. Then when we look at this difference in lateral line patterning, we find basically the same pattern of association, that the strongest peak is here at these two length polymorphisms. If we look at neuromass number, this is a little hard to see, but the number of neuromass per body segment also varies. That also shows a strong association with the same region of the haplotype. And this suggests to us that there's a pleiotropic mutation, that within the small, basically 1 kb region of the haplotype is where we see the strongest association for plates and the neuromass, suggesting pleiotropic mutation. Interestingly and surprisingly, actually, no other traits mapped. So for those 120 other traits that we looked at, nothing ETA haplotype, suggesting that they are due to linked loci outside of the haplotype. We did find evidence for a little bit of linkage of an additional mutation within the haplotype that affected both lateral plates and lateral line. So this is sort of these are the genotypes at this one of these length polymorphisms or indels. And so again, CC fish have more plates than CL fish, have more plates than LL fish. But if you look at the SNP sort of on the right-hand side of the haplotype, in these CL heterozygotes at this locus, if they're LL at this SNP 1213, there's actually a reduced number of plates. So there seems to be sort of a linked mutation that modifies the effect of this mutation at the CMV 770. And we also see this for the dorsal ventral, this neuromass patterning. We don't see it for the neuromass. So basically the summary is that we find evidence for a pleiotropic mutation somewhere in this region of the haplotype that affects the plates and the neuromass, but there is a linked mutation within the haplotype that sort of modifies this major mutation. Okay, so the conclusions of this part of the talk and sort of where we're going, that we still questions. That's what I love about science, is you sort of answer one question and it opens more questions, keeps you in business. We find evidence for the pleiotropic effects of a single mutation in ETA, but I haven't shown you what the causative mutation is, so we have been working really hard on we have a good candidate. We think it's one of these indels, and Carlos Ramirez is a PhD student in the group, and he is conducting the CRISPR-Cas9 experiments to sort of recreate this mutation and see what the effect is. So stay tuned for that. And then we really only measured morphological traits. And we know that there's super strong selection for this locus in freshwater. So this ETA low or freshwater haplotype is under very, very, very strong selection when the fish go into freshwater. And we still don't know sort of what is the source of selection on this locus. It could be also multiple sources of selection, because this 16 kb haplotype has been maintained actually over a long period of time, when you would think recombination should have sort of chopped it up. So these other genes in the haplotype are associated with immune function. And so a master's student in my group, Stephanie Walker, is investigating whether there might be immune functions associated with this this haplotype. So it's sort of new work that we're getting into right now. Another major conclusion is that this QTL cluster on chromosome 4 that we see is not just due to the pleiotropic effects of EDA. We can't just explain all these 41 phenotypes by EDA. There's clearly linked alleles that are also contributing to freshwater adaptation. And so a question that I've been thinking about for a long time is, how do you build these sort of clusters of linked alleles? How are these clusters built? What's neat is that chromosome 4 and chromosome 7, which both show these clusters, turn out to be fusions that have occurred in the lineage leading to Threespine sticklebacks. So they're unfused chromosomes and sort of sister groups, but they've been fused in the Threespine stickleback lineage, which either brought together sort of adaptive alleles that were already existing into linkage, or created a region of low recombination in which these sort of adaptive clusters could be built. And Zuyao Liu, who's a PhD student in my group, has been working on this question and sort of looking at signatures of selection within the fusion. So again, stay tuned for future work there. Okay, so I'm just going to turn to a second part of the talk now. I mean, really, I've been presenting sort of pleiotropy as a sort of a positive force in adaptation, that you sort of bring together these adaptive alleles that can bring together different traits that sort of make a good freshwater fish, for example, that allow it to adapt to parasites, to predators, sort of. So pleiotropy is a good thing. But sort of the classic literature, so Fisher proposed that pleiotropy would be a constraint on adaptation. And so the idea was, so this is just a figure from his book, so where mutational size is sort of dependent upon the number of characters affected. So large mutational size would be sort of more pleiotropy, small mutation size is basically not pleiotropic. And so in his model, the probability that a mutation is favorable is very dependent upon mutational size. So less pleiotropic mutations are more likely to be favorable. And the idea is that if a mutation affects many traits, it could be beneficial in one axis of selection, but detrimental in others. So the probability that pleiotropy is basically synergistic is low, and the probability that pleiotropy is antagonistic, sort of in selection of acting on these different traits in different ways, is much higher. So really, Fisher predicted pleiotropy would constrain adaptation. Okay, but as Christian and I were discussing earlier, empirical tests of this are quite rare. And so Diana Renison, when she was a postdoctoral fellow in my group, wanted to address this question on sort of a genome-wide scale. Does pleiotropy facilitate or constrain adaptation? I should just mention Diana is now an assistant professor at the University of California, San Diego, running her own group. Okay, so how did Diana set about trying to address this question? Diana's insight was that repeated evolution might provide an interesting test of this idea. And so we know that there's, and I've talked to many people about this today, evolution repeats itself, at least at the phenotypic level, right? We see the parallel or convergent evolution of similar forms in sort of placental versus marsupial mammals, or the classic example of parallel or convergent evolution of very similar forms in independent lakes, Great Lakes in Africa, of these cichlid fish. And so convergent and parallel evolution, first I just want to define my terms here, is the independent evolution of similar phenotypes in similar environments. And when I use this term convergent, I generally mean they evolve from a different common ancestor. And so marine mammals provide a nice way to illustrate this. So for example, the elephants or manatees, their closest relative is an elephant. And so they have convergently evolved a marine lifestyle from dolphins and killer whales, so they have a very divergent common ancestor. But dolphins and killer whales would be an example of parallel evolution because they've evolved from the same common ancestor. So it's just a terminology difference. The real point I want to make here, though, is that repeated evolution is most often interpreted as evidence for natural selection. So when you see the same phenotypes evolving in very similar environments, it suggests that this is not just due to drift, that there's actually a role for selection, and that the traits that you see are actually adaptations to these different environments. Okay, so sort of parallel and convergent evolution provide us as a sort of a test that a locus is under, or something is under selection. And we, of course, also see that evolution can repeat itself at the genetic level, and there's these very spectacular examples, for example, of convergent evolution of light pigmentation in lizards and mice, and that these sort of evolutionary changes have occurred through changes, for example, in the same gene, and sometimes even the same mutations can lead to very similar phenotypes in very different organisms. And I think these spectacular examples might have led to a little bit of a bias in the literature and people's thinking that sort of things that when we see the similar phenotypes evolving, it always should be the same genes and the same mutations, which I think many of you we've talked today that is definitely not always the case. But when it is, we might ask the question, why is evolution repeatable at the genetic level? Why might the same genes or mutations be used when the same phenotypes are evolving? And so one answer is just there's only one gene or mutation that can produce that phenotype. So there's only really one way to do it, which I think is probably quite unlikely. The other is that there may be many genes or mutations that can make the phenotype, but some are less pleiotropic and have fewer fitness constraints. So this would sort of be Fisher's prediction that if a mutation doesn't have pleiotropic effects, then it can is not constrained and can contribute repeatedly to the evolution of the same phenotype. It could just be that some genes have higher mutation rates. And there's actually a beautiful example of this in stickleback, where the repeated loss of pelvic spines is actually due to mutations in this independent mutations in this gene called PIDX1, which is in a fragile region of the genome, and so it's just more mutable. It could also be that there's some genes or mutations that are just in regions of low recombination, and therefore sort of can be selected. Okay, so again, Diana set out to sort of address these questions using two different cases of parallel evolution of freshwater species pairs. So these Benthic-limnetic pairs, which are found in 5 independent lakes in British Columbia, and then we've also looked at these lake stream pairs, which we've examined 16 independent lake stream pairs. So I'll give you just a little bit more details of these systems. So these Benthic-limnetic pairs are found within a single lake where you have a benthic species that lives in this sort of near-shore environment that's adapted to feeding on benthic macroinvertebrates, and this limnetic form, which is living in the open water of the lake, which is adapted to feeding on zooplankton. And so we had genomic data from 3 independent Benthic-limnetic pairs. This was genotyping by sequencing data. And in total, Diana could estimate FST between Benthic-limnetic pairs from about 35,000 genome-wide SNPs. For the lake stream pairs, we're sort of evolving along a similar axis. So stream sticklebacks are adapted to feeding on more like benthic macroinvertebrates, so sort of more like a benthic-y species, whereas their parapatric lake partners are adapted to feeding on zooplankton. Again, we have genomic data from 16 independent lake stream pairs. This is actually double digest rad-seq, so slightly different data. And again, we had about 68,000 SNPs, and Diana estimated genome-wide FST. So the first pattern is that there's repeatable genomic differentiation in the Benthic-limnetic pairs. So these are just FST plots across the genome, and you can see these really striking sort of similarities in where you see high FST across these three different pairs. Incredibly striking, actually. Across the 16 lake stream pairs, we see sort of not so striking patterns. So the sort of overall levels of FST are not the same. And sort of these sort of really repeatable patterns across the genome are not the same. But as I'll show you, we do find some repeatability of regions of genomic differentiation. So just to summarize, this is all a windowed analysis, 50 KB windows. And so Diana identified 172 windows that were parallel, meaning they were differentiated in 2 or more pairs. So sort of at the top 5% FST in a population comparison. And then she had just over 400 windows that were only differentiated in one pair. So about 30% of the windows were parallel. In the lake stream data, we saw a similar sort of percentage of windows that were differentiated. So 377 windows differentiated in 2 or more pairs. Some even differentiated sort of, 9 to 10 of the 16 pairs. And then about 2/3 of the windows were differentiated in only 1 pair. So basically, we have our parallel windows and our non-parallel windows. And we're going to ask then, do these differ? So the parallel windows should be sort of those windows that are under selection across, suggesting there's sort of consistent selection across the replicates. So do these differ in the level of pleiotropy? And so we measure pleiotropy in 2 ways. So of course, ideal way to measure pleiotropy is you knock out every gene in the genome and you ask how many phenotypes it affects. And you'd have to do that under lots of different environmental conditions. It's sort of crazy. I mean, people have done these sorts of studies in yeast or in worms, but we had to use proxies. So we had 2 proxies. So the number of QTL from this big data set that David Marquez and I had generated, and gene connectivity. So Diana used RNA-seq co-expression network analyses and looked for genes that were sort of, if a gene is co-expressed with many other genes, this is taken as a proxy that it's more pleiotropic. It's basically interacting with more genes. And there's some work to suggest that that's actually a good proxy by doing functional studies. And these proxies have different sort of pros and cons, and so our idea was like, let's just use a couple different ways to look at this question and see if we come up with the same answer. We also wanted to know if these windows differed in gene number, in recombination rate, or in mutation rate. Okay, so to get to the answer, so the first result is that the parallel windows actually contained more QTL. So on average, parallel windows in the lake stream comparison or the Benthic-limnetic comparison contained about twice as many QTL as those non-parallel windows. And these were significant differences in both comparisons. We also did a lot of things like looking at only outlier windows. So if you just compare parallel outliers versus non-parallel outliers, we see the same effect. We sort of use ranked FST instead of this just coarse parallel versus non-parallel analyses, and we see the same pattern that if a region is parallel, it's likely to have more QTL than expected, or sort of more pleiotropic, which is sort of contrasting to the prediction of Fisher that these parallel regions would actually be more constrained and therefore sort of less pleiotropic. When we look at the connectivity data, we see a very similar pattern. So again, parallel windows contain genes with higher connectivity than the non-parallel windows. This was not quite a significant difference in the Benthic-limnetic pairs. One thing I want to point out, though, is sort of the non-parallel windows tend to have the most pleiotropic loci, right? So sort of on average, they have lower pleiotropy, but sort of these highly pleiotropic loci seem to be more enriched in the non-parallel windows. So I wanted to now just summarize what we found rather than showing you all of the plots. So parallel windows have higher QTL and higher connectivity, but the non-parallel windows seem to be the most pleiotropic. There is no difference in gene number, so we can't really explain that difference. Recombination rate, so parallel windows in the Benthic-limnetic pairs have a lower recombination rate. But that in those systems, there's very high gene flow. This is in contrast to the lake stream systems where gene flow is actually quite variable. And so if we just took the outlier windows, so these are all regions of low recombination, then there's no difference in recombination rate between parallel and non-parallel windows. So we can't explain sort of our results simply by sort of recombination rate. And there's no difference in mutation rate between these windows. So really, our explanation is that these parallel windows seem to have a higher amount of pleiotropy and therefore seem to be contributing to adaptation more often. So to conclude, pleiotropy, what we would say, does not always seem to be a constraint, and rather maybe these intermediate levels of pleiotropy, especially pleiotropy that's synergistic, so having effects sort of that take you in the same direction might actually be adaptive. And sort of to tie this back to the first part of the talk, these old alleles, so this Eta freshwater marine haplotype is actually millions of years old and has maintained a standing variation. And so you have you've had repeated bouts of adaptation to freshwater throughout the stickleback's evolutionary history. And so it could be that sort of these old alleles have been tested by selection, and so they've been tested, they have this sort of good pleiotropy or synergistic pleiotropy, and so then they're maintained in the system because of that. And so that may be something a little bit unique to sticklebacks that means that sort of negative or antagonistic pleiotropic effect mutations are sort of weeded out, and these synergistic pleiotropic effects are the ones that remain. Okay, so with that, I will stop so that there's some time for questions. I'll just thank again David for the work with the QTL database story, and then Sophie led this really beautiful work on association mapping, pleiotropic effects of Eta, Diana on this genome-wide analyses, and then we had help from our colleagues at the University of Washington in our collections for the big association mapping study in Lake Washington. So yeah, with that, I will stop and gladly take questions.